Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College, Autonomous, Mahabubnagar. Today we will continue our lesson, Mineral Nutrition in Plants. In our last class, we have seen the criteria of essentiality, how the elements are classified as essential elements and what basis, and total how many essential elements are there. So we discussed that out of 60 elements present in the plants, which we come to conclusion on basis of the ash analysis, a total of 60 elements are required or are present in the plants, but only 16 of them are essential. On what basis? Arnold and Stout has proposed a criteria of essentiality, and it has 3 points. So we will say what are the 3 points. The element must be essential for normal growth and reproduction, and in the absence of that particular element, the plants do not complete their life cycle. The second one is the element cannot be substituted by any other element, and the third one is the element must be directly involved in the metabolism of the plant. Okay, so based on this essentiality criteria, uh, there are 16 essential elements identified so far. Okay, so they are again classified into macro elements and micro elements based on their requirements. If the requirement is 1000 microgram or above per gram of the dry weight, then they are called as macro elements. And if the requirement is 1 to 100 micrograms per gram of the dry weight of the plant, then they are considered as micro elements. The macro elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. And the micro elements are iron, manganese, zinc, boron, copper, molybdenum, and chlorine. We saw as last class that what are the sources of these macro elements, and what are the functions they perform within the plant body, where they are the components or constituents or activators or what else they are, or how they are performing their role within the plant body. Then we also saw the deficiency symptoms of different macro elements. Today we will see micro elements. Okay, so micro elements. What are different types of micro elements? These micro elements are also known as trace elements, as the requirement is in traces, very less concentration they are required. So what are the different micronutrients? Iron, manganese, zinc, boron, copper, molybdenum, and chlorine are different micronutrients. So we will see one by one. The first one is iron, and iron is it constitutes 0.01% of total concentration in dry tissue of plant. And the source of iron is either ferric or ferrous ions, that is Fe3 plus or Fe2 plus ions. But Fe2 plus ions are physiologically active in plants. And you all know that iron is abundant in acidic soils. Then the functions, regarding the functions, iron is an essential for the synthesis of chlorophyll. Even though it is not one of the constituents, it is essential for synthesis of chlorophyll. And it is important constituent of cytochromes as well. Cytochromes, we saw their role in cyclic and non-cyclic electron transport systems, and they're also the constituents of catalases and peroxidases, uh, and these catalases and peroxidases again they play a role in photorespiration. Okay, then the iron is also one of the important component of ferredoxin. Ferredoxin is also one of the electron carrier. And nitrogenous enzyme, nitrogenase enzyme, it is also a component of nitrogenase enzyme. So these are the functions of the iron within the plant body. And the deficiency is extensive chlorosis in young leaves. What is chlorosis? Yellowing of leaves or disorganization of chlorophyll is chlorosis. So extensive chlorosis in younger leaves is observed as a deficiency symptom of this iron. Then uh, the next one, or the one more deficiency symptom, is the chlorotic leaves do not die at once, as in other deficiencies induced chlorosis. They remain on the plant as a terminal cluster of white leaves. See, you can see here, this is the terminal cluster of white leaves formed. Okay, so here it is a chlorosis, here intervenal chlorosis. You can see here only in between the veins yellowing of leaves has been observed here. This is chlorosis. Total leaf uh, becomes yellow. This is intervenal chlorosis, and here the terminal cluster of white leaves you can observe here. Okay, so these three are the deficiency symptoms of iron. Okay, iron is present in 0.01% of the dry weight of the plant. Okay, so the next one is manganese. Manganese constitutes 0.005% of the plant's dry weight, and it is observed in the form of manganese ions, that is Mn2 plus ions. And manganese is deficient in alkaline soils because it is converted to unavailable forms in alkaline soils. And what are the functions of the manganese? It is an activator of enzymes of respiration and nitrogen metabolism. For example, malic dehydrogenase, an enzyme of Krebs cycle, requires manganese as an activator. It is also involved in photosynthetic split of water, that is photolysis of water. In the presence of manganese and chlorine, only the photolysis of water takes place. So it is very important to photolysis to happen. Then it is also connected with the synthesis of chlorophyll. So these are different functions of manganese. Then deficiency symptoms. What are the deficiency symptoms? The symptoms of manganese deficiency are chlorosis and necrotic spots on older leaves. Again, chlorosis means yellowing of leaves. Okay, so chlorosis and necrotic spots, dead localized regions. Necrotic spots are nothing but dead localized regions. Okay, and ultimately they form into dry spots. And the manganese deficiency occurs in alkaline soils because in alkaline soils manganese is converted into unavailable forms. Okay. So their availability, we can uh, they are sufficiently available in acidic soils but non-available in alkaline soils. And in leguminous plants, seedling shows necrosis. And some of the diseases caused by these manganese deficiency are gray speck of oats, marsh spot of peas, speckled yellows of a sugar beet are due to this manganese deficiency. So these are about the functions, availability and deficiency symptoms of the micronutrient, that is the manganese. Okay. Next one is zinc. Again, zinc is uh, present in 0.002 percentage in the dry weight of the plant. Okay. What are the sources? Zinc is widely available in the soil in the form of Zn2 plus ions. And if the pH of the soil increases, the availability of the zinc also uh, in, increases. Okay, it decreases. The availability of the Zn plus 2 ions decreases with the increase in the pH. So, zinc deficiency is common, uh, especially in citrus orchards and alkaline soils. So, what are the different functions of these zinc within the plant body? Zinc is involved in the synthesis of indole acetic acid, which is a natural auxin, and it is also an activator of several enzymes such as alcohol dehydrogenase, pyridine nucleotide dehydrogenase, and hexokinase. And the deficiency symptoms are uh, the intervenal chlorosis. You can is clearly observe here intervenal chlorosis of older leaves starting from tips and spreading to the margins. And in severe cases, the leaves become small, internodes are shortened, resulting in stunted growth. Leaves become distorted. They change their shapes. See, you can observe here the leaves are distorted leaves. You can see here. So distorted, smaller in size, and they form cluster as rosettes. Okay. Some of the common uh, disease symptoms caused by the deficiency of this zinc are little leaf disease, example, little leaf and rosettes of apples and peaches. White bird of maize is a zinc deficiency in maize, and it is characterized by stunted growth. Reduced flowering and fruiting is also observed. And mottle leaf of citrus, sickle leaf of cocoa are also due to zinc deficiency. So all these are deficiency symptoms of zinc, and the main important function is involved in synthesis of indole acetic acid, which is a natural auxin. So this is about the micronutrient zinc. The next one is the boron, and boron is observed in the form of borate ion, that is H2, BO3 minus, and present in small quantities in the soil, and that is 0.002% of dry weight of the plant shows the very little concentration of boron, that is 0.002% of boron is present in the total dry weight of the plant, okay, and observed in the form of borate ions present in very small concentrations in the soil. And what are the different functions performed by this boron? So the functions are it is important in plant metabolism, that is boron is involved in translocation of sugars. Sugars are translocated in the form of borate solvents, okay. So it plays very important role in translocation of sugars and also in synthesis of DNA in meristems. These two are the functions of boron. And what are the deficiencies caused by this boron deficiency? Boron deficiency causes death of shoot tips. The leaves show thick coppery texture. You can see here the leaves show thick coppery texture. These leaves are showing the thick coppery texture, and they are also showing curling. See, you can clearly see here. Okay, so thick coppery texture and curling are seen in the leaves with the boron deficiency, and flowers do not form, and root growth is stunted. Uh, in storage organs, disintegration and browning of internal tissues may be seen. See, you see here, browning of internal tissues is clearly observed here in this picture. So these are the deficiency symptoms, and it causes some diseases in some plants. We'll see hot root of sugar beet and the corky core of apple, water core of turnip, and top sickness of tobacco growth are due to this boron deficiency. Okay. So this is about boron, and the next one is the copper. So This copper is it constitutes 0.0006% of the dry weight of the plant. Excess of copper is toxic to plants. So then the source of copper is it is observed in the form of Ce2 plus ions. Excess application of phosphate fertilizer reduces its availability. Its deficiency is common in fruit orchards, okay, where we spray a lot of phosphate fertilizer. The functions of this copper are copper plays important role as a catalyst. Okay, it is a constituent of several oxidizing enzymes such as ascorbic acid oxidase, cytochrome oxidase, tyrosinase, phenolase, etc. It is present in chloroplast, that is in plastocyanin, which is again an electron carrier, which plays an important role in both the cyclic and non-cyclic phosphorylation or electron transport systems. Okay, so it is a constituent of plastocyanin present in chloroplast. Then these are a few functions of this copper, and the deficiency symptoms are uh, young leaves show withering of tips, which leads to loss of leaves. Plants may wilt even when water is supplied. That is, even though if water is supplied to the plant, they show wilting due to the deficiency of this copper. Okay. And exanthema is a disease of fruit trees in which gum exudates accompanied by dieback and glossy brown blotches on leaves and fruits. You can see here in the diagram the exanthema of fruit disease, which is caused by the deficiency of copper. So these are the deficiency symptoms. So copper is observed in the form of Ce2 plus ions. Excessive application of a phosphate fertilizer reduces its availability, and functions are constituent of several oxidizing enzymes such as ascorbic acid oxidase, cytochrome oxidase, and phenolase present in plastocyanin. Plastocyanin is a copper-bearing electron carrier which is present in chloroplast as an important electron carrier in photophosphorylation. The deficiency symptoms are young leaves show withering of tips, plants show wilting even when water is supplied, flowers do not form, and root growth is stunted. It causes exanthema of fruit trees where gum exudates accompanied by dieback and glossy brown blotches on the fruits can be observed. Okay, this is regarding copper. Next one is molybdenum. Molybdenum. Uh, this is uh, this is present in a very minute quantity comprising of 0.00001% of its total dry weight. Sources of molybdenum are it is readily observed from soils with a high pH. It is available in the form of molybdate ion, that is MoO4 minus in the soil. Okay, it is short supply in acidic, sandy, and granite soils. And what are the functions? It has an important role in nitrogen fixation and nitrate reduction. And it is an activator of nitrate reductase enzyme. And deficiency of this molybdenum leads to drop in the concentration of ascorbic acid. Deficiency symptoms are uh, chlorotic intervenal mottling. You can see here intervenal mottling. The leaf structure is changed completely. This is called uh, mottling of the leaf. So intervenal chlorotic intervenal mottling of leaves is observed as a deficiency symptom of this molybdenum and leads to chloroplast disorganization. You can See here yellow patches which are seen on the leaves. These are disorganization of the chloroplast and it causes whiptail disease of cabbage and cauliflower. Whiptail disease of cabbage and cauliflower due to the deficiency of the molybdenum. Okay, and sometimes plants do not become chlorotic but develop several twisted young leaves. Twisted young leaves are also the deficiency of the molybdenum. The next one and the last one is chlorine in the micronutrients. Okay. Chlorine is about 0.01% in the dry weight of the plant tissue. The source of chlorine is available as a Cl minus ion, that is chloride ion in the soil, and the functions are uh, it remains in the form of a Cl minus in the plant without becoming the structural part of organic molecules. It stimulates the split of water, that is involved in photolysis of water. Already discussed that manganese and Cl minus ions are very important for photolysis of water, which supplies the lost electrons to photosystem 1. Okay, then it it is essential to maintain target pressure, that is essential for target control associated with the sodium and potassium ions. Okay, that is closing and opening of this what you call stomata is also dependent upon this target control, which is associated with Na plus and K plus ions. Okay. So the deficiency symptoms are roots become stunted, leaves wilt and fruiting is reduced, and leaves show chlorotic and necrotic spots, and bronzing of leaves is also appear. So here you can see here bronzing of leaves. The leaves shows a bronze patches over them. Okay, that is bronzing of leaves. So this is about the micronutrients. So what are the micronutrients? Micronutrients are those which are required in trace quantities, that is 1 to 100 microgram per gram of the dry weight. And how many micronutrients are there? A total of 7 micronutrients are found, that is iron, manganese, zinc, boron, copper, molybdenum, and chlorine. And we saw their sources, their functions, and various deficiencies caused by these micronutrients in plants. Okay, this is all for today. Thank you.